of De-Extinction. This idea that you can bring an organism back to life. What Colossal is doing is super interesting, but they just exaggerate everything. With the direwolf, they didn't even come close to creating a direwolf genome. They wanted to take an Indian elephant and put the mammoth genes in it, and then have an actual Indian elephant mother give birth to it. At the end of the day, it would probably end up in a zoo somewhere, and people would pay a lot of money to go see the mammoth. They're doing this to manipulate funders and get money.
Jon Perry is the scientific illustrator behind Stated Clearly, the YouTube channel that turns genetics, evolution, and biology into crisp, gorgeous animations. Nearly 600k subscribers, 50 million views, with early classics hitting 5 to 6 million each. He's been debating Dr. Michael Levin on a hot question: is biology cognition all the way down? Levin says we should call cells intelligent. Jon says that muddies the science. So he built a Lego maze to show how seemingly smart behavior can emerge from simple physics. What counts as intelligence, life, and learning, and why does it matter? Are cells intelligent? Drop your opinion in the comments below. And remember to subscribe if you want to stop your tap water qualifying for Mensa and keep your reality stated clearly.
There was this paper published by this Japanese group. They found that the slime mold is really good at solving mazes, and they said this might be some primitive form of intelligence, and the media exploded with that.
Right.
So I redid their maze of the original Japanese experiment. I rebuilt it with Legos. I filled it with sand instead of agar. They had agar in them, and they had food. The slime mold will grow towards food and will reduce its body to where the food is. Water, it goes towards gravity. It falls downhill. So I'm like, well, I can use that as my attractant, just gravity. And sure enough, it could solve the same maze, did it faster even. It's not the type of thing that we call intelligence in cognitive research. He's got a definition of intelligence that tap water fits.
Jon Perry, you had a disagreement recently with Dr. Michael Levin, and I think it created a really interesting conversation online. Talk about what happened.
Well, kind of rough to start out this way, but yeah. So I was doing an experiment. I had filmed an experiment. It was actually years ago. I filmed an experiment. I've always been bothered by people saying that slime mold is intelligent, because if you look in the long history of cognitive research, what we've been doing, the main project is trying to tease out instinct. So evolved instinct from active intelligence, from actual problem solving. So that definitions vary quite a bit in the field for what intelligence is, but one that almost no one would disagree with would be that intelligence is the ability to perceive that there's a problem and then reason through a possible solution. So you see this happening in a huge diversity of organisms. At first we thought it was just like, oh, apes can do it, and dolphins, and then people are like, octopus can do it. And we've actually been able to tease out through very careful experiments that some insects are doing this as well. You even have insects that will teach each other how to do stuff. It's quite amazing. So intelligence goes way further than we thought it did, even in that strict definition. So there's this huge, this goes back, really it goes back to Darwin, maybe even before him, where people are trying to tease this out, instinct versus like active intelligence. So when people start saying that cells are intelligent, it's been, I would say probably right after World War II, we started saying that cells are capable of a type of learning that's similar to conditional learning, because we found out that there's these gene regulation things, even in bacteria, where they can detect certain sugars in their environment, and then they start upregulating the enzymes that can digest that sugar. And then when there's not that sugar, after a while, they will stop producing those enzymes. So we learned that right after World War II. Some people called this learning. Other people didn't like that. So anyway, you fast forward to 2020, there's this, or no, it was 2000. There was this paper published by this Japanese group. And I really think it was a throwaway sentence at the end of their paper. They're like, They found that the slime mold is really good at solving mazes. And they said this might be some primitive form of intelligence. And the media exploded with that.
Right.
So I had filmed, I think this was in 2020. So during COVID lockdowns and stuff, I was going crazy in my house. And so I'm like, I'm going to do a little experiment and show that this is BS. So I redid their maze of the original Japanese experiment. I rebuilt it with Legos. I filled it with sand instead of agar. They had agar and they had food. They had food that's luring the slime mold to two spots on the start and the end of the maze. I'm using gravity to lure because that's what I know. The slime mold will grow towards food and will reduce its body to where the food is. Water, it goes towards gravity. It falls downhill. So I'm like, well, I can use that as my attractant, just gravity. And sure enough, it could solve the same maze, did it faster even. So I was sitting on that for a while because there was such a strong anti-science movement during COVID that I didn't want to accidentally feed.
Right.
So I sat on that forever. I didn't really know how to frame it either because slime mold really is doing something special. It's using the same physics. It's actually using just flow dynamics inside of its body to solve this problem. It's the liquid being passed from the two spots that are eating the food. That's what's actually solving the maze. That liquid going back and forth between those two sections. It's using really the same principles as water flowing downhill. But it's exploited that for survival. And that's significant. That's really interesting. But that falls in the same category as unevolved behavior and instinctual behavior. It's not the type of thing that we call intelligence in cognitive research. So I published that, and the first video I mentioned, I tagged him in it because he talks about this all the time. He talks about slime mold intelligence quite frequently. He talked about it. I don't know if he talked about it in his TED Talk. I thought he did, but someone told me afterwards that they don't think he did. I'll have to go back and look at that. But other people have talked about it on TED. It's been brought up multiple times on TED. And that's one of the places where it exploded. It's just,
Right.
Slime mold is intelligent. Slime mold is intelligent. And it just blows everyone's mind. And then they get excited about it.
On Joe Rogan as well. This is the original Paul Stamets clip, right? That went like super viral. And I swear like every bro science guy kind of saw this one clip of Paul Stamets on Rogan like 10 years ago and echoed it to like 10 of their friends. So this is the clip we're talking about.
Right. Yeah. So, and I love that guy. I love the.
Me too.
He's so fun. Like, so I grew up in the Northwest, and there's a lot of scientists I know that are like him. They're like ex-hippies turned scientists, and they're awesome, and they get excited about things, sometimes too excited, and they get the way he talks about things publicly sometimes. But it's like and this is what Michael Levin is doing too. He gets really excited, and I understand it, but the name of my YouTube channel is stated clearly. I care a lot about clarity, and I actually started my career. My actual training at the university, I wanted to be a scientific illustrator. That's what I wanted to do. So I studied biology and I studied illustration. And then I got into a graphics design program as part of that. And I had to take a bunch of marketing classes. And I actually ended up, like right out of college, I was doing a marketing job. I got hired as the art director for a company called Win International, which was a multi-level marketing company.
Wow.
And they were totally scamming everyone. And they would use these word tricks, and it drove me nuts. I was getting ready to quit. I had been there for a couple of years. I was getting ready to quit, and then the company failed. So I'm like, oh, I guess I don't have to quit. But I was so disgusted by just marketing and marketing terms, and I'm like, I'm done with this. I ended up getting a job at Oregon State University. I would work with professors to they would take their classes and they would move it over to online classes. And at first, Oregon State University was just filming lectures, and students who were paying top dollar for their online courses were angry about that. They're like, I'm watching a recycled lecture. This could be done so much better. So I was in charge of trying to figure out how do we translate these lectures, compact them. I could work with a professor and take a 45-minute lecture and turn it into 5 minutes, and it's still the same information.
Right.
I was really liking that job. That led to my YouTube channel and everything. But the reason I brought up all of this is I just have this like deep, like visceral hatred for word manipulation.
Yes.
And that's what I saw happening in this slime mold is intelligent. Like we have, this betrays the public understanding of this word, which is, the common understanding. And it also betrays the operational definitions that have been used for over 100 years in this field of cognitive research. Now, that doesn't mean that we have to respect old science. I mean, scientific innovation, it's always vandalism. We ruin the history, the science of the past when we make a real breakthrough. But this, I don't see, there's no value added. There's a little bit of value added, I think. One of the things he's pushed for and his colleagues have pushed for is the way we used to think of intelligence is you have cellular behavior, and that gives rise, certain types of cellular behavior give rise to cognition, and certain types of cognition give rise to intelligence. And so you have that hierarchy. And one of the things that people have been pushing, some of his colleagues have been pushing, is to say that those types of cellular behavior that give rise to cognition, we should call those basal cognition, to draw that link and really drive home. In some ways, this is just a vibe shift. We want to change how we're talking about things. We're not really innovating anything. It's a vibe shift. But even from a vibe shift, you can see things from a new angle. You can notice insights that you didn't realize before. And I think that that's valuable. But what Michael Levin and other people are doing, they're just saying everything is intelligence. So the basal cognition is intelligence. And that's just like, I really care about clarity, especially I care about not confusing the public with kind of markety jargon.
It's definitely an interesting one. So I've been trying to drink less alcohol. And honestly, it's had a pretty big impact on my health. I'm sleeping better, I'm exercising more, and my business is growing. The data suggests that people are drinking less alcohol than ever, which overall I think is great, but I'm still looking for a little treat at the end of the workday or a hard workout. And that's where Sentia comes in. Sentia is a 0% alcohol drink that feels like you're drinking something special without any of the downsides. It's created by neuroscientists and it's designed to give you that relaxing buzz. The same kind of feeling you get from alcohol, but without the hangover, brain fog, or impact on sleep quality. And it tastes absolutely delicious. They already have a GABA red berry, which is perfect to mix with a tonic or orange, or GABA black, which is awesome with Coke. Personally, I've been really enjoying the GABA gold with Fanta lemon on ice.
That's actually genuinely delicious. Like, I'm not even play acting there, it's actually just really, really good.
So you might be wondering, how does a drink with 0% alcohol give you that buzz? It almost sounds too good to be true. Well, this is the really cool neuroscience part. The secret is GABA, the brain's natural calming agent. And the different GABA flavors have different cognitive effects. Sentia's founder, Dr. David Nutt, has published over 500 independent research papers and written 34 books on neuropharmacology. What Dr. Nutt realized was that we can harness the same calming, relaxing effects from alcohol without any of the side effects. I genuinely think this could be the future of drinking. And they're also just releasing their first beer and stout, which as an Irishman I am dying to compare to Guinness. Sentia are offering my viewers 10% off their first purchase, and I genuinely could not recommend it more. I think even if you're looking to just substitute your one gin and tonic on a Tuesday with Sentia, the cumulative health benefits over time will actually be very noticeable. So check out Sentia, again 10% off at checkout. I definitely see your point, and I see what you're saying. I brought this up to Dr. Levin on our podcast because I was kind of thinking, well, do we need more words? Do we need a different word for the types of kind of behavior that seems intelligent? It seems like learning memory, but they're cells. Do we need a new word for this? And he was very adamant on like, no, I think different words would actually do it a disservice to what it is actually doing, and that he fully believes. And I think I would more lean towards his perspective after my podcast with him in terms of thinking about intelligence as a continuum rather than binary. And to push back a little bit on the value add, I think that is value add in terms of rethinking about intelligence as not this thing that either an organism has or doesn't have, but that there is degrees of intelligence and it exists on a very broad continuum. And that is a mental model reframing that I definitely had throughout our episode. And I think that does help me think about it's expanded the definition for sure, but it seems appropriate to me. It seems like it is the correct word. And he's made very good cases to me that it should be intelligence the word intelligence that we're using when we talk about things like cells or xenobots or anthrobots or other organisms or even bacteria, or he would even go more radical into more sort of primitive systems. And I don't know if I fully agree with all of them, but that that value add is there to me in terms of redefining it in a way that makes sense to me, that it's not binary. It's not a thing that someone has or doesn't have, but it is a continuum. Do you think intelligence is a spectrum?
Well, yes. I mean, we have, again, we go from cellular behavior to, and certain parts of that really you could call basal cognition, and that is a term that he uses in his papers. So he'll say one thing in one point and another thing in other cases. I mean, he knows how to use this vocabulary, how the field has been using it. I think that basal cognition is, I mean, it's this perfect tentacle down into cellular behavior, which is what he wants. But one one thing that one of the arguments I've made here is that, you know, there's a spectrum between, as you're building a kitchen table, if you're woodworking, there's a spectrum from the raw lumber to when the actual kitchen table is finished, right? Or when you would start to call it a kitchen table. So it is a spectrum, right? There's always a spectrum here, and we have to deal with all this stuff with. Actually, this might be a better case. I'll talk about how we define life too, because this is actually a multi-billion-dollar question. We've really screwed up with probes that we built for NASA because we didn't have a good. I mean, you want to say you don't want a binary. These terms are always fuzzy at borders. Most terms in biology are fuzzy at borders. A species is, you can tell that a dog and a cat are different species, but there's some really weird things that happen when you talk about a dog and a coyote or a dog and a wolf. They can interbreed and so on. We are very much used to having fuzzy boundaries on definitions, but that doesn't mean we just say that everything's a dog, or, you know, all lumber is a kitchen table. You get to, I mean, it gets absurd at a point, right? So you really do have to draw a line in the sand somewhere, and it's going to be arbitrary, and people are going to argue over the exactly where that line is drawn. But he's got a definition of intelligence that tap water fits. If you like, there's a paper he did in 2024. I'm not sure what he would say about this, and I want to talk to him about it. But if you just read that paper, it seems like tap water fits, because he's just saying that if something can reach a goal, and we say that something has a goal according to him if we see it doing something many times in a row. So tap water, we keep seeing it. It always goes into the sewer. So that's its goal. Its goal is to go to the sewer, and it will. And so he says that something is intelligent if it can get, if you block it in one way, it'll find a second way to get there. Well, the tap water, it is determined to get to the sewer either through the toilet or through the kitchen sink. So it's going to fit his definition of intelligence there. And I just think that that's too, like, first of all, it betrays our common understanding of intelligence. Secondly, it betrays what everyone's doing in the field for what I would say is no added value. I mean, I get that you disagree with me on that, and that's fine. But
On
I just,
water, I would love to hear his response there. I'm nowhere near as intelligent as him, and using intelligent in a way that you'd be happy with there. I don't know what he would respond to in relation to water. But you bring up an interesting point. Like you say, the boundaries are fuzzy. Like, I don't know that there are boundaries. You know, like I really do, after speaking to him and speaking to a number of other people on the podcast, really do think it's useful to think about biological systems entirely in continua and entirely in spectrums. And that's an interesting model that I think frees you up to think in different ways, that it isn't a categorical difference. I mean, there's so many examples of this, right? In psychiatry, we have labels for diagnoses and medication, and that can be useful, but there's also a very big movement at the moment to rethink, are these labels really useful? Because they are arbitrary cutoff points in a threshold, and everything is spectrums and continuums. And I really do believe challenging a binary model can help with experimentation. Like, is water intelligent? It sounds ridiculous. I'm not going to say it is, because you're right that just seems too absurd. But I'm also coming around to the fact that possibly bacteria are conscious and possibly plants are conscious, and that is also a shift, a radical shift in what I would have said a year ago. And I think it's because having these conversations with people has made me realize that binary categories distort our reality. They distort our thinking into saying, oh, obviously that thing is not a thing. I think it's very easy to say that, where he will say, look, you have to do the experiments, right? It's not useful to say something obviously doesn't have a characteristic. You have to do the experiments. And I was looking into the paper he talks about on the different characteristics and features of that. But I agree with loads of what you're saying, but I disagree. I think the value add is that we get out of binary thinking. Would you agree with that? I think not binary thinking is good in biology. Like, I don't know anything in biology that is binary, and maybe you can
the normal field of intelligence right now is not binary. It's very you have these. There are very different types of intelligence that we've discovered, but we have what we're trying to do, and where parts do get binary is where we're saying, is this an evolved instinct? Is this conditioned learning? Or is this something that we would call intelligence? So we have these cutoffs, but they are very, like, so you can do experiments to figure out how an animal is doing its behavior. How does this bug remember its pathway home? Some of them are using a trail. They're excreting things and they're leaving like breadcrumbs for themselves. Some are counting, and we've done experiments like kind of horrible experiments to figure this out. They cut the bottom parts of the legs off. I can't remember what bug it was, what insect it was, but it was one where they said, "Oh, it doesn't feel pain. We don't think it feels pain." So they thought it was fine to do this, and it would lose its way because its steps aren't as big, and they were able to tease out in its mind what it's doing, or it's somehow
That's
counting
really
these steps. Maybe it's counting time, or maybe it's actually counting individual steps. We don't know. But so we're able to do all these very careful experiments, and you do need, in order to do any sort of experiment and learn anything from it, you have to know what is confirmation of your hypothesis and what is not. You need to know what your hypothesis is.
Right.
And so that's why you will see people using slightly different variations of their definition of intelligence, and you'll see that something from one paper doesn't really cross over to another one, but there's a whole community of people, and they're playing the same game, and then all of a sudden you have someone who suddenly sounds like he's making a crazy breakthrough when what actually happened was it's a word game. That's what bothers me. That really bothers me, and they get tons of attention for it too. It's what I call concept doping, and Michael Levin is not the only one who does this, and but he does do it more than most people do. He does it.
Do you think because he wasn't even making claims in our conversation, he wasn't even making claims about consciousness, and I actually felt like he was conservative on labels personally. I'm not saying he doesn't do this maybe in areas that I haven't seen, but I actually thought he was conservative in throwing out labels. What he seemed really concerned about was testing these things in experiments. Like he really wanted to get away from labels and binary thinking, and he said a label has to be operationalized in a particular way, but what he cares about is doing the experiments, being the engineer, which I think he would appreciate, you getting the actual Lego maze out and doing the experiment, right? That's not armchair philosophizing. You went and did the experiment. I think he would appreciate that. But I actually thought that he could have thrown out more labels than he was. How do you think about, have you looked into his Xenobots and Anthrobots? Because I think these things are remarkably cool. And I think he could have made more claims about them than he has in terms of what I see them doing. And I'm like, that's crazy. What are your opinions on them? Have you looked into these?
Yes. So I saw you asking him in quite amazement, like, you were asking him, what was it like when you first saw these things come to life, these creations of yours come to life? And I'm curious, like, what it is, like, what do you think these Xenobots are? Like,
so I might have said the words "come to life." I think I used the words "move." So they're clumps of cells that seem to have strange. I'm going to use the word emergent, but he doesn't like the word emergent behavior. That they find themselves in an environment that evolution didn't intend, because they're departed from the rest of the body and they're isolated in this controlled system. And these clumps of cells can move around a petri dish with the flagella on their skin, on their cells, and pile together other clumps of cells which start to act together.
Mm-hmm.
What they are, I don't know. That's it seems to me like a form of synthetic life. I don't. They have altered gene expression, which, I mean, you're the expert on, and I really wanted to get your opinion on this. It's like, why the hell do they seem to have such altered gene expression where they seem to be able to detect vibrations? Like, he put little sound dishes underneath the petri dish, and it was seemed to be able to receive that sound signal, alter its behavior, and then return to normal. And it's like it's a clump of heart and skin cells from a frog.
Mm-hmm.
You have Anthrobots healing neurons, like getting two parts of a neuron and stitching it back together. Like, these are strange behaviors. I don't know if behavior is appropriate, but you could say they're behaviors of clumps of cells that don't
behavior,
but that don't really seem like the inert matter that maybe someone might assume a clump of heart and skin cells would be.
Yeah.
But I'm curious if you disagree with anything there.
Well, okay, so what he's doing is he's creating what everyone else calls cellular aggregates. People have been doing this for, I don't know. I think some of the early papers on cellular aggregates are like in the 70s or so. I might be wrong. It might be more recent. It might be like the 80s or 90s, but people have been messing with cellular. So how you do this is you take tissue and you pull cells off of it, and then you put them together in new little clumps. And there's lots of different ways to do this to make cellular aggregates. And you can take them all from one organ or from multiple organs and combine them. What we usually use them for is making models. People don't like to, it breaks our hearts to be torturing rats all the time in labs, right? So people have found, I can make organoids out of these cell clusters. So I can take heart tissue and I can test things on it. I can take lung tissue and test things on it. And some people have found really cool little tricks where you'll take like if you want to test, if someone's having lung issues, they got a lot of mucus in their lungs, it's a child, and you want to know if they have a genetic disorder that's causing this. Well, we know of some genetic disorders that can cause this. It can cause the cilia to form wrong, but we don't know of all the genetic disorders that can cause that. So what you can do is you can grab some of their cells and through some manipulation techniques get them to form a little cluster, a little ball, and develop their cilia, and then you can study how that cilia swims without having to do some crazy invasive surgery on the patient. So that, I mean, this is experimental. I don't know that that's
Cool.
actually being used in hospitals, but
Really cool example, though.
people have been doing this forever. We call
Right.
them cellular aggregates. His big innovation was to start calling them robots. That's the big breakthrough. It's a word change. Like
Well,
we've already been
there
doing
was
this.
quite a lot of computational optimization, right, in terms of to getting it to do this thing, running millions of simulations and optimizing the configuration, just to separate, because the listener might get into their head that this is literally just an arbitrary clump of cells, but they ran very large models running different morphologies of these cells and kind of found an optimal configuration, right? I don't know if, maybe I could be wrong here. I don't know if anyone had taken it that far, but maybe that's wrong.
No, he's using what he's doing, and I don't know that anyone else has done this. I know that people are using software to help them. So like with the cilia thing, people develop software to analyze what the cilia are doing, so they can try to distinguish different disorders, and measure how well it's the cells are moving. But yeah, so his what he added to the science there was doing. So you can just tinker. Just think of this. You can just play around with Legos and put them together in different ways and see what kind of shapes you get. Or you can model those Legos first. And in this case, it's easier to model them first because it's pretty hard. It's hard to manipulate individual cells, right? So you're saving a lot of time if you can simulate things efficiently and then use what you learned from your simulation to go actually build the cellular aggregates, right? So that is an incremental increase in that's a genuine contribution to science. He's saying we can use these computers programs to help us build cooler cellular aggregates. But the big thing that got all the headlines was he started calling them robots, and people are like, "Oh, this is synthetic life. This is a new life form. It's replicating." Um, the form of replication that he has, there's no information transfer from parent to child, and he talked about this when you pushed him on it.
Yeah.
There's no information transfer from parent to child. It is exactly the same. If you have a snowball bouncing or rolling down a hill, it's going to kick up other little snowballs.
I was thinking that's how you think that's what's happening.
Yeah, it's just so these cells can stick. All of these cells can stick to each other, but they have to be pushed against each other first to stick.
It's my understanding that it makes rough copies of itself, though. It's not, and it's not exact. He said the copies are not exactly the parent, but it does make it makes better than you would expect by chance if it was just arbitrarily collecting the clumps of cells into a pile. It forms the same configuration that can move, and there's no real reason it's not stochastic, right? I think I asked him that as well, and he said no, it's not. At least it doesn't seem the kinematic replication isn't stochastic because it is creating rough copies of itself with, of course, errors. But that means it's not a snowball per se.
This comes from the behavior of the cells themselves. So once the cells stick to each other, and once multiple cells are sticking to each other, they'll form kind of an outer shell, and they'll develop their cilia, and they'll start swimming. So all you have to do is push them together in a large enough clump, and then they'll form a clump that then also starts swimming around and bumping into other cells and causing them to form clumps. But what he did is he designed one using his software. He found one that's going to make clumps even better because it's got a little kind of like a Pac-Man shape to it.
Right. Yeah.
So that one's really efficient at forming clumps. But the clumps that it forms they're just going to be the round clumps, or
they're
kind of
random. Okay, I
clumps.
thought the clumps it made were also Pac-Man.
No.
Interesting.
So that there's no information transfer from parent to child. This is very similar to like snowballs bouncing down a hill, kicking up other snowballs. It's the same as crystal seeding. It's slightly different than crystal seeding. It's very similar to how fires spread. So a spark can cause a new fire, but there's not information from the parent fire transferred to the offspring fire. So you don't, we had, and this kind of brings us back. I want to talk about the definition of life. Like, what is life? We had, we went through this argument in Origin of Life research back, I want to say it was in the 60s and 70s with Sidney Fox. Sidney Fox found a form of replication very similar to what Mike Levin's talking about. It was budding. So he developed this, he called them proteinoids, and they're these little particles that when you put them in water, they start forming clusters. And when those clusters get big enough, they will branch off, and then you've got two orbs. And so there's this, he's like, this is replicating, this is alive. In his actual papers, he was very careful not to say that this is life, but like at conferences and stuff, he was saying, I've created life in my lab. And he was telling people this at the bar after the conference and so on. So it caused this big stir. I mean, it was probably years of someone, it took a long time for people to figure out exactly what was wrong with what he was saying, because it looks really cool. Like, you're an Origin of Life researcher. You want to be able to solve this problem. It looks like he just solved this, or a major part of this problem. Like, look, we have replication. Isn't that what we need for evolution? And then I think it was, I can't remember who it was who pointed it out, but it was basically shown that this is just seeding. This is, again, similar to snowballs reproducing. It's similar to crystal seeding. It's similar to a fire reproducing. This does not give us, this is not, I mean, it's interesting that this happens, but it's not interesting for the origin of life, in that there's no information transfer. This is not the thing we were hoping it was.
Right.
So
makes sense. And good example.
We've gone through all these debates before. And so it's frustrating to see it come up again and make huge headlines. And it's like, come on, people, like, you must know about this. This is just, it's everywhere. It's in the field. It's well published, well discussed. Like, why are we repeating this? But we were talking about fuzzy definitions earlier. One of the fuzziest transitions, I mean, is this transition from non-life to life.
Right. Somehow
chemistry gave rise to life. And one of the arguments, and this was Carl Sagan who really pointed this out, is that it's going to be very helpful for us to have a definition, an operational definition. It's not going to be perfect, but we need an operational definition that we can work on. And this became super apparent after, I think it was the Viking probe on Mars. I did a video about this called "What is Life According to NASA?"
Talk about this. Yeah, I don't know. This is interesting.
They spent billions of dollars getting a probe onto Mars, right? And they had a bunch of tests for life on that probe. And the tests came back positive. Like, we found life on Mars. And this was like back in the 70s, was a huge, obvious headline, right?
Could you give an example of what one of these tests might be?
These were fairly simple chemistry tests that they were testing for types of molecules that we thought could only be produced by metabolism, or could only be easily produced by the metabolism of a cell.
So DNA, RNA, these.
Yeah. So basically, it's stuff that cells excrete when they're doing their stuff. So basically, we thought that we found bacterial feces, right? Molecules that you wouldn't expect could just form on their own without enzymes, without life causing them.
Metabolism, yeah, makes sense.
So we had tests like that on this probe, and then someone's like, "Actually, I can simulate a non-living system that makes those same molecules." And they're like, "Oh crap, we did not do a good job foolproofing our test before we spent billions of dollars launching it into space," right? The equivalent of billions. I think back then it was not billions, but it would be the equivalent now. So NASA put together this big conference, and it's fairly controversial. because they came out with a very black and white definition that life is a self-sustaining chemical system capable of Darwinian evolution.
Yeah. Which
Now,
is the
if you want
definition
to
you
make
kind of hear
tests,
often
what's that?
enough. That's the definition you hear often enough, right?
Right.
Yeah.
And that is a fuzzy, that's fuzzy because, like, well, what is a chemical system? What what is real Darwinian evolution? How open-ended does it need to be? Because you could have very limited Darwinian evolution. So there's plenty of fuzziness in that category. Does a virus count? By that definition, most people would say yes. And actually, by that definition, it opens it up to some very interesting things. Genes would be considered alive by that definition, some people have pointed out. And
Right.
some people have pointed out that your grandma does not count as life because she's gone through menopause. She's no longer
Yes, that
capable
is
of.
the other menopausal women don't self-replicate, right?
So
That's
there's
the law.
weird, or a man with a vasectomy or whatever. So there's a bunch of weird problems in there. And then, of course, I can't reproduce. I'm not capable of Darwinian evolution on my own because I'm an individual, but it's through mating and so on. So there's tons of weird stuff and fun things to argue about in there. But it does make it's like you can design your test, and even for some simpler definition or some other definition, you can design a test. But that if you're satisfying this definition, that everyone's going to be impressed. Like if you found something that is doing this on Mars, like you can crack open the champagne pretty comfortably without having to, take it back later. And I would argue that this is just very important that people can get behind an idea like this and know what we're testing when we're testing it, because right, you have to know what you're looking for when you're doing an experiment. Otherwise, it's actually not totally true. You can just observe things. So, like Michael Levin's observation that these what he calls I think it was like the human bots. I don't remember what he calls those.
The Anthrobots.
The Anthrobots.
The trache made out of the tracheal cells.
Yeah. His observation that they healed a scar on a tracheal cell, that was like an observation. He didn't hypothesize, if I put these on tracheal cells, they'll get healed. That was an observation. And these types of things are very important in science. We just observe what's happening. There's tons of papers where people are documenting what animals do in the wild. You don't know ahead of time. You don't think you know ahead of time. You just go watch. So a lot of what he's doing is that, and that is a legitimate, solid contribution to science that he's doing there. But it's it is very important to know that he didn't design it to cure neurons and then it cured neurons. He's watching it. It's curing. It does this thing, and in vitro, and in a petri dish. I think if you were to put those cells in a human, well, maybe if you made it from their cells, then it would. It'd be interesting to see what happens there. But so what he's doing is interesting. Like he's using people have been using these cell aggregates up until now. They've been using them mostly. I mean, some people have just been playing around and tinkering and building fun things, like this little spinny ball that can teach you about your cilia, right? Or your patient's cilia. But for the most part, we've been building what we call organoids with them. So little models of an organ, and then you can test drugs on that little model of an organ without hurting a rat, and it's cheaper and it's nicer because we don't think that cells suffer as much as entire rats. But again, there's a consciousness question, which you seem to be very interested in.
I am.
And you're right. Michael Levin doesn't make consciousness claims. He makes intelligence claims. And those are very different claims.
And he could, to be fair, I think it's where I think maybe he deserves some credit in that. I think he could reasonably make that claim in that it seems like they have subjective experience. So there's definitely some definitions of consciousness that it does fall into. But I feel like he has been more, like he's not posting on X every day saying he created consciousness or life, you know. Right. And I think a lot of other scientists would have. And you know, I think it's just everything about the way he presents himself, and you know, he has like a 720p webcam that he like you know refuses to
Yeah.
upgrade, and like he is quite a humble man, I believe anyway, and that's why I think based on what he has achieved and what he has accomplished, it seems to me that you know in a random, if you were to sample his amazing successes in 1,000 people, how many of them would have claimed that they created conscious life? And I think it's a very large percentage. I think he's understated his achievements, and his paradigm shifts in bioelectricity. You know, I think it's just so interesting and so important, and I think he's generally very understated based on what he's accomplished. But I really do hear your argument. I understand the perspectives, and I'm curious for your conversation with him, because he'll be able to respond a lot better than I. I'm not here to represent
Yeah.
Dr. Levin.
Yeah.
I couldn't, and
Yeah.
I absolutely can't. So I'm curious to hear what he says.
And I don't want to pick. I feel like I've kind of I've ended up picking on him too much, because he does do a lot of this, what I call concept doping. Like you know, the thing that got the headlines wasn't a giant innovation in cell aggregates. It was calling them robots. That's what captured the headlines. But he's doing solid basic research where he's moving these fields forward one experiment at a time. He's doing what you would want a good scientist to do. He
Right.
just has this marketing thing on top, which uh, which I find like I am hypersensitive to this, having the history that I've had in marketing and just seeing this this type of manipulation. But he's not the only one who does it. He's not the worst who does it. And so I feel bad for picking on him too much, because also when he gets these big funding grants because of his good marketing, he's using it to do he's not embezzling money. He's using it to do basic research, and he's tinkering and he's figuring out cool things. He's done, I would say, what I would say is the most important thing that I understand of his work that he has done is he has proven that it is valuable to talk about bioelectricity outside the world of neurons, right? We've been talking about bioelectricity in brains and the nervous system since forever. We've thrown lots of money into understanding electric signaling in the brain, right? But most people have been ignoring, not everyone, but most people have been ignoring electric signaling in cells. We focus on molecular signaling in cells, between cells.
Yeah.
And he's saying, look, no, the same stuff is happening. What happens in a neuron is happening in normal cells. They are using bioelectricity to solve all sorts of problems. And I think that the experiment that he did that showed this the most is there's people have been messing with planaria. They cut the heads off of it, and someone figured out, I don't remember who it was, someone figured out how to use some molecular tricks to get them to grow two heads instead of a head and a tail. And they were doing this manipulation. It was quite complicated. They were hitting the system. Whatever it was that they were doing, it was pretty complex. He showed that you can do the same thing much simpler if you attack the electric signaling system, if you just focus on that. So he put some drugs in there that jam the electric signaling, and it did the same thing in a much easier way. So it was like a simple small molecule that he threw at it, instead of, I think people were using like a morpholino or something to block a gene transcription. And he did the same thing with a simple small molecule. And that's like eye-opening. It's like, oh, we should, instead of focusing on blocking genes, we can just focus on interrupting what they're doing at a different level, and we can get the same result. And this is, I mean, there's people that do that. Most drugs, of course, don't block genes. Most of our interventions don't work at the gene level. But he showed in this world of developmental biology, we focus a lot on the gene level.
Yes.
And he's like, yeah, you can actually ignore that in some ways and just block the electric signals.
Which is huge. Like I remember, like I did 4 years of developmental biology without hearing the word bioelectricity. It was just exhaustive ridiculously named genes one after the other in really cool and really sophisticated systems, but the word bioelectricity was not mentioned once. It was not in the curriculum. It was not a feature of the system. It was not a factor that implicated how the
Yeah.
genes were expressed. And basically, the first time I had heard of it, of bioelectricity even being a relevant factor in developmental biology, as someone who's studied neuroscience and is reasonably well read in the periphery of these topics, is from him. I am,
In
and
neuroscience?
yeah.
Oh, you guys didn't study electric signaling in neurons.
Oh no, sorry. Like in develop
Okay.
as basically. No, of course, neurophysiology, of course. No,
Like,
I mean in the developmental mechanism of embryos, it was all genetics
yeah,
the whole way down. I
yeah.
am, of course, in
No,
neurophysiology,
no,
of
you're
course.
right. You're right. And this
Yeah.
is this is a big, very important shift that
Yes.
I mean, and he doesn't try to take full credit for it. He'll tell you the pioneers in trying to do this, because people tried to do this a long time ago, but when the structure of DNA was revealed by Watson and Crick, and then people started realizing how cool this is, all of our attention and money it was just dumped into genetics. Like
Yeah.
that's the cool thing, especially for developmental biology and so on. So there is this very important things got neglected, and he's pointing this out. And that, like, first of all, he's done experiments to show this, and that's what you have to do to really make your case. People aren't going to listen to you until you prove it. And he did it with this. He's like, I can do what you were doing easier with these smaller molecules, these simpler molecules. That's a big, like, slap in the face. Like, oh, okay, well, he's going to get people jumping on board because of that. And this is very
Yes.
important. That should not be like dismissed. His
Yes.
push here is super important.
Right.
It's going to lead to a bunch of new discoveries for sure.
For sure. I'm glad we were able to come
Yeah.
to one agreement on Dr. Levin's work.
Yeah.
Great news. If you were thinking of getting your own genetics test done, then Nucleus Genomics, the leading consumer company for genetic testing, is giving my audience 10% off their first test with the code EVAN10. The whole process is super smooth and simple, and you'll get a really intuitive breakdown of everything that you need to be aware of within your DNA. Your breakdown will include risk scores for different conditions that you might be predisposed to. Oftentimes, with this information, you'll be able to take proactive steps for your health. For example, in my test, I found that I have 2 rare mutations that predispose me to a condition called hemochromatosis. This is a condition where my body can't really break down iron very well, and hence it starts to accumulate, which can cause a bunch of negative side effects like inflammation and brain fog. It means that I should get my blood tests done every single year, and I need to be specially aware of my iron levels. But even more importantly, this information is also relevant for my dad, who's always commented about having high iron levels. So your genetics test is of course not just for you, but also for your family, who might discover something important and actionable as well. Get your genetics test done today for 10% off using my link in the description. I really could not recommend Nucleus Genomics more. You're far more read than me on things like gene expression. So I'm really curious to get your explanation for why, when you have a clump of frog and heart cells in a dish, how do you start getting these altered gene expressions that seem like an adaptive mechanism to its environment, and sort of feed into this idea that maybe it's intelligent, maybe it's conscious, because it seems to be responding to its environment and then engaging in some sort of goal-directed behavior. I mean, put all of those things in quotes as undefined and ambiguous and fuzzy boundaries and everything.
Yeah.
But talk a little bit about that, because I found that interesting and I don't fully understand it.
So our cells, any cell. Do you ever spend much time? Do you happen to have a microscope at your house? You ever
I
just look at
don't, but I want one.
Yeah. This was another thing I did during COVID, as I bought a microscope and I started looking at pond scum to keep myself sane. And it's amazing when you just watch at these little single-celled organisms. So, some of the things you find in there are multi-celled, some are single-celled. Sometimes you have single-celled organisms that are way bigger than your multi-celled organisms. it's
It's
so
something I
fun
really
to.
want to do, actually. For ages, I haven't, and I've kind of forgot that I'm an adult and could just purchase a microscope. And
Yeah.
that is something that I would actually deeply love to do.
Yeah, even the cheaper ones, like mine was $100 at the time. I think they're like $300 now because of, whatever. but the yeah, it's not that much, and it's
Yeah.
hours of entertainment, and it's yeah, I mean, you can also just go look on the YouTube channels, like Journey to the Microcosmos is a really good one for that. There's one. Oh my gosh, I'm sad that I'm forgetting his name. Microbe Hunter is my favorite channel. This guy, he's just very calm. He's a biology teacher. He has a high school classroom that he teaches, and he just does a bunch of microscopy videos on the side, and he's so good. And he
Love
teaches you
that.
how to use your microscope and everything. But yeah, when you start watching these cells, you'll see all of these fascinating behaviors, and they behave in what seems like, yeah, they're acting like animals, and it makes you want to say that they are intelligent, and it takes careful experiments to distinguish, like, what are they doing. There's even conditioned learning, according to, you know, I think this has been pretty well confirmed now, that cells are capable of conditioning, Pavlovian type conditioning.
Yeah.
That people were debating that for a while, but I think it's pretty well established now that cells can do that. That's one of the behavior, cellular behaviors that we would say is basal cognition, right? But how they do it, in many ways, and the ways that we really understand, is that they change gene expression. So cells have all sorts of receptors on the outside of their body. So the receptors that you'd be familiar with are the ones on your tongues, or the ones in your nose that you can actually, you have receptors in your nose that are physically reacting to molecules that you breathe in. So that molecule hits this receptor, and the shape of that molecule causes the receptor to bend, and that causes a response that gets triggered, goes all the way to your brain, and you sense a smell. But inside, you can have them trigger all sorts of things. So these receptors can be wired up, you could say, inside the cell to trigger all sorts of different behavioral changes in that cell. One of those behavioral changes is to just switch gene expression. So every one of your cells has your entire genome in it. Each one of them does, except for red blood cells. A couple others that don't have genomes at all. They expel their genome as they mature. But most of your cells have a complete copy of your genome, at least one copy, and most of the genes are turned off. They're just bundled up in proteins and so on that stop them from being expressed, stop them from being used. You have, when the receptor on a cell is triggered, when the right receptor is triggered, it will cause a chain reaction that opens up one of these genes and makes it start expressing the gene that was previously covered up.
Right.
And we first learned about this in bacteria right after World War II with, I think it was like I think it was, yeah, lactose.
The lac operon.
Yeah, the lac
I hated
operon.
those lectures in college.
Yeah.
God damn.
Yeah, well, I think they teach them in the wrong way, because it's super
Yes.
fascinating. I mean, it's this beautiful.
I'm sure it
feedback
is. Yeah, but I didn't like it on a lecture slide. That would be the kind of thing that I would need a clearly explained animation.
Yeah, I
for.
should do.
Yeah.
I should do a Stated Clearly video on that discovery, because that discovery changed everything. Like we understand the mechanisms. This is so cool. We understand how cellular memory works at a mechanistic level. That's awesome. Well, one of the ways. I mean, there can be other ways too. But so when you take a cell and you put it in a new environment, it's going to be all sorts of different receptors are going to be triggered in weird new ways, and so it's going to change the gene expression. That's not. It's like we see a new emergent behavior. If I tie my dogs together on a leash, they behave differently. right? If I have two dogs, and I can have them leashed to me, or I can have them free, or I often just leash them to each other, and they're way easier to handle, but they have a lot more freedom. But they behave totally different. They start to behave totally different because they're tied to each other. This is an emergent new behavior. Same animals, I just change the situation a little bit, and they behave differently. And so this is what cells are doing. You pull them out of their normal environment, and they're going to start behaving differently. In a lot of ways, that's what Michael Levin is doing. And you might think, well, that's not that interesting. But guess what? You can tie dogs together and create an engine for a sled, right? That's cool. So by tinkering around and what he's doing is he's putting these cells in new situations. It's causing new genes get turned on and turned off. He
So
doesn't
what
know ahead of
controls
time.
that exactly? Because that's maybe the bit that's I'm uncertain on. Like why would he said that when he looked at the genes that were being upregulated, they seemed to be a cluster around hearing, and this was a huge surprise. Like why the hell like could this clump of cells actually hear? And that's when they stuck the little speaker under the dish, created the sound waves, and then it seemed to change the behavior of the Xenobots in the dish. So like why
Yeah.
on earth would you get upregulation of hearing genes in a situation of a clump of cells composed of heart and skin cells? You know, like what is that? Do we know why that would occur specifically? Like, is that a bit of a mystery?
So that paper I have not read. He might explain it in that paper, but I think what he's doing is, again, having not read that paper, I would assume that what he's doing is he's putting these cells in a new situation, because this is what he does in other papers that I have read. He's putting a cell in a new situation, observing it, and then documenting the observation. So he's observing, I put these cells in here. For some reason, they started expressing hearing genes, and I could register that, so I'm documenting it. It's like a wildlife biologist going out and seeing what lions do when they're not at the zoo. So that's very important research. From what you're telling me, if I were doing that experiment, if I saw, oh, it's expressing hearing genes, now when I subject it to sound, it behaves differently. I mean, what I'd be curious is that because of the hearing genes, or is it because cells respond to sound? Cells do respond to sound. Sound is a vibration. Vibrations cause all sorts of things to happen differently inside of a cell. If you shake it, if you take a dog and shake it, it'll start behaving differently, right? But so it's not, again, I'm just getting what you just told me just now, so I don't have enough information to say. But reading that paper, you might find out. He might have really nice theories as to why he thinks that what's happening is happening, or he might just be documenting, look, we did this and this is what happened. And
In general, would
both,
you? Sorry, go ahead.
Both are valuable. Just documenting
Right.
what's happening, that's valuable. Knowing why it's happening is more valuable, but that's something that usually comes later,
Right. In general, would you expect the genes that are upregulated to be advantageous for that organism's survival in that moment? Like, is that the mechanism that is causing particular genes to be upregulated over others? Or like how sort of, I understood a lot of what you said there, but I wasn't sure on exactly like how does it choose what genes to be upregulated in what scenario, and what's doing the choosing? Is it random? Is it stochastic? Is it based on like some sort of back and forth information between the organism and the environment? Sort of talk around that a little bit.
It's going to be like, there's a video online where an owl falls in the water and starts swimming. I don't know if you've ever seen this, but
No,
owls aren't swimmers, right?
I've seen them run though, and they're terrifying when they're running with their big
Yeah.
legs.
Yeah. Like, if you put an animal in a new environment, it'll start using what it's got to try to it's going to start using what it's got. And the owl has the ability to go like this. It's really good at going like this. And so it does that in the water, ends up being slower, ends up being fairly effective. It can kind of swim, and it can get to the edge, and it can in some cases, if there's a nice exit for it, it can get out of the water. these cells have evolved to maintain homeostasis. They've evolved a bunch of behaviors. When you put them in a new environment, they're going to do new stuff. Whether or not it's good for them it's kind of a coin toss.
Okay.
I think most of the things that they're going to do in a new environment are going to be like, they're going to be trying to stable. Well, there are some weird exceptions here. They're going to be, if you have disabled their apoptosis system, which cells want to kill themselves all the time. When a cell finds itself in a new environment, when a human cell finds itself in a new environment, it tries to commit suicide because it thinks that it thinks something is wrong. It thinks something is wrong. It detects that there's something wrong, something unusual is happening. And in most cases, when something really unusual is happening, it's either infected by a virus or it's become cancerous. So cells in multi-celled organisms like us are super prone to apoptosis. They're trying to unalive themselves, as the kids say on YouTube these days. But a lot of times for these cell aggregate experiments, you have to disable that in some way. And there's different ways to do it. You can use stem cells, which aren't super prone to doing that, or you can use certain cancer cells, or you can delete that gene. There's a bunch of ways to do it. But once you've done that, you do see that it does seem that cells will behave in ways that tend to be fairly good for their survival. So they are behaving in ways that are adaptive for their survival.
Right.
But they have a very limited capacity. It's what did evolution already give them to do that? There's not a lot of they're just highly limited. But you're going to see all kinds of novel behavior, just like when you tie animals together for the first time. You'll see them do all kinds of weird stuff, and then they'll start to normalize and get, they adjust to the feedback, and they can start to actually go in a straight line without tripping on themselves.
Right.
It's very similar. You have a system that's trying to self-correct in a cell. So
really cool. Well explained. I think we beat that Dr. Levin discussion to death. So there's other topics that I really wanted to.
I feel bad because I feel like I'm picking on him too much.
No, it's interesting It's really interesting conversation. It's new. It's weird stuff, right? And it brings up a whole bunch of interesting conversations, I think. And that's what generally why I just wanted to start on that, because it was topical recently, and I saw loads of people getting engaged with loads of opinions on both sides, and I just think it's important conversations as stuff like this is happening more and more, as we get more technology to be able to create new forms of synthetic life. Again, we put so many air quotation marks on all of these different labels, but stuff like this is going to happen more and more. So important to sort of have the conversations around them now, and what it means, and is it ethical, is it moral? It's a whole other section we didn't get to. Talk about Colossal, Biosciences, and Direwolves.
Yes. Yeah, this is a lot nicer. I would much rather pick on Colossal than on Michael Levin, because
let's do it.
Because it's a whole they have a full-fledged marketing team. I think Michael Levin is good at coming up with the best word to use that's going to get headlines, and he does that. But it's like intuitive for him. He just kind of does it naturally. This is like, you've got a team of people that are really actively tricking their audience.
Yes, talk about this maybe from the start. Direwolf De-Extinction.
Well, first off, what Colossal is doing is super interesting. They are doing interesting things, and they are moving science forward, but they just exaggerate everything to a level that's, if anyone agrees with me that what Michael Levin's doing is a little bit sketchy, like, that's nothing compared to what this company is doing. There is a word that became popularized called de-extinction. It got popularized through TED Talks mainly, but it was already being thrown around. This idea that you can bring an organism back to life. You can, and it's also sometimes called, resurrection of an extinct animal. The this was popularized with Jurassic Park, and even in Jurassic
Yeah.
Park, when you watch Jurassic Park, that it's like, oh, well, it's not the full dinosaur DNA. We had to plug it up with frog DNA because there was holes and what we had. If you read the book, I don't know if they talk about this in the movie, but in the book, in Michael Crichton's book, right? He talks a lot about how they're not full-fledged dinosaurs. They're actually dinosaurs mixed with frog DNA and so on. So there's this idea that you can bring an animal back to life, and it doesn't have to be necessarily 100% that animal, but you can bring it back to life, and it's kind of there's a level where it's good enough, and we're going to be impressed, right? If you were to make a mammoth, a giant elephant with huge tusks and big furry fluffy hair, most people are going to be impressed, right?
Yeah.
And that's going to be awesome. And especially, it's going to be impressive if a lot of those genes came right from the mammoth genome. And we have mammoth genomes that are pretty good because we've got some of these that are preserved in permafrost, and their genomes are pretty well, they're degraded, but we've figured out our ancient DNA techniques are getting pretty good now. We can sequence a genome of an extinct animal that's pretty well, not perfectly, pretty well. In that case, and this is where it really got popularized on TED, there was a TED talk about this. They wanted to take an Indian elephant and put the mammoth genes in it. So just edit the genome, put so that it looks like a mammoth genome, and then have an actual Indian elephant mother give birth to it, and so on. And the idea is that that's going to be really close to the original. And that's pretty stinking cool, right?
If any of that is true.
Well, yeah, and animal rights issues aside, but
yeah, which is a big aside because I still don't know where a mammoth goes. I had a conversation before where their habitat was Siberia, and I don't know if anyone has asked the Russians if they want to take on a bunch of mammoths. Otherwise, they're going to be.
That actually is something that Colossal has been very good at. They're working with indigenous communities in Canada, and I think they're working with people in Russia, and they've actually secured, like, this is where we would let them go.
Wow.
So they've got permission, and that's not easy. One of the things that happens all the time one of the conflicts that happens fairly frequently in Canada and in Hawaii is you have like this headbutting between native cultures and Western science. You want to build a telescope on a sacred mountain and so on. That's a big issue in Hawaii right now. And Colossal has proven that if you're just nice from the start and you don't pretend that you deserve this, you can work with these people, and they're not irrational. They hate being told what to do, and they're tired of being, this colonial mentality. But if you treat them with respect, they'll be like, yeah, that's cool. I want a mammoth. That's awesome. So Colossal has done a good job with this. But people argue, well, it's going to change that ecosystem, and it's going to be an invasive species now.
Seems like the biggest question mark for me is you're reintroducing something that it isn't really a mammoth, right? It might look like one, but it's not a mammoth in possibly important ways in an ecosystem that's, I don't know how different, but assumingly quite different. Yes. So we've no idea. It's like we talk about uncertainty when you're dealing with very complex systems, and like an ecosystem is a highly complex, delicate system. It kind of seems like mad scientist mode completely to take an organism and then just stick it in an environment and even remotely predict what would happen.
Right. It's very, we cannot track the moving parts. When you disrupt an ecosystem, the consequences are not not accurately predictable.
As much as I want mammoths, again, I think all of the science lovers obviously we want these things to exist. I think it's the saddest fact in the entire world that species have gone exist and humans have caused species to go exist. But I'm just incredibly skeptical of this idea of creating sort of elephant. And we can really go into the details on how mammoth-like this thing will be, and how
Right.
the direwolf is, and how
Yes. Yeah.
the thylacine is, and that's where I think we can really give some good genetics. But it's
Yeah.
just expecting we can predict what it's going to do in the environment seems a bit crazy to me.
Yes, it does. And I think probably what would end up happening in real life is it would end up being at a zoo. That's
Yes.
probably how
Yeah.
this stuff is going to shake out. Maybe we'll end up releasing it. But even if we did release them, it would be a soft release. It'd be in a limited area. It'd be a limited range,
Be sectioned
and
off.
so
Yeah.
on. So I think you can do it responsibly, or relatively responsibly. But at the end of the day, it would probably end up in a zoo somewhere, and people would pay a lot of money to go see the mammoth. But this brings us to the other question you had: is how mammoth would it actually be, right?
Yeah.
First of all, let's just pretend, and this is definitely not what they're doing. Let's pretend that they're trying to get the genetics identical. The genetics are identical to a mammoth. There's a bunch of other things. There are maternal effects. There are epigenetic effects that happen that are caused by the mother, caused by the egg
Yes.
cell. So within the egg cell, there's a bunch of stuff in there that shapes the gene expression of an organism as it's developing. So, I mean, you know about mitochondrial DNA, I'm sure, which is in the egg cell, not the sperm. I'm not sure if they're trying to put mammoth mitochondria in there as well. Maybe they are. But you're definitely going to have maternal effects of all sorts, meaning that probably the first couple generations are still going to have a lot more. Even if their genomes were identical to a mammoth genome, they would still have maternal effects, making their phenotype be more like
Yeah.
an Indian elephant, or just modified in weird ways we can't predict because of maternal effects. So that's one thing, but that's
It's a
kind of
good
minor.
point.
You
It's
could
probably
breed
a
that out.
lesser known good point.
Yeah.
Yeah.
After multiple generations, that would fade because maternal effects fade through time. So if you were having these things breed with each other,
Right.
that would fade. And you'd have to have some genetic diversity to stop, inbreeding side effects and so on. But that's not with the dire wolf. They didn't even come close to coming to creating a dire wolf genome. There are ways to say that what they said is okay. So this word de-extinct, if you look at the definition of that, which was published in a paper by, I can't remember the name of the group. It's like an endangered species group that published a paper on it, and they defined it. In that paper, they said, we don't like our definition of this. It's got problems for all of these reasons. But they defined it as the creation of basically a proxy for an organism that is extinct. So any proxy. They didn't say how close it has to be. They didn't say anything about its genome. They just said that it's acting the way the extinct animal acted. So Colossal picks up on that. I'm like, oh, the bar is super low. We don't have to we can use this word if it's a proxy. I mean, you could actually say that a wolf is a proxy for a dire wolf. I mean, there's no boundaries here. So let's play.
Yeah, speaking of operationalizing definitions as we have for so long, like surely proxy has to have a better definition and more rigorous boundaries. It could mean anything.
Yeah. So, but they did what they did that's interesting. So they did stuff that's interesting, and it sucks that it gets buried in the scandal that they also committed here, because their researchers are doing, again, it's like they're doing this to manipulate funders and get money, and they're not embezzling it. They're using it. All of their projects are expensive. I don't think anyone's embezzling money there. That's not what I'm saying. I'm not saying that they're committing fraud. They're just tricking the public, and they're maybe there are types of fraud that some lawyer would find in there, because it wouldn't be fraud. I think it would be some type of negligence, of investor misleading through negligence or so on, by just exaggerating your claims the way that they're doing. But
Hard to find a venture-backed company that doesn't really do that implicit
yes,
in their model, though, right? It's like
yeah,
that's what venture-backed
Tesla,
companies really do.
yeah,
They all, it's
Tesla
yeah,
gets sued all the time for for. I think they keep getting sued for negligent manipulation or something like that.
yeah.
Maybe sometimes fraud, but yeah, it happens. Lawyers know how to define these things. I don't want to throw out the wrong accusation and then I get sued, but no. What they actually did, partly interesting. They went and they found genomes that are fairly nicely preserved. They found a genome in a tooth that was good enough to where we could get a lot of information from a dire wolf, and they found information enough of the genome and other specimens to get a really nice rough outline of the dire wolf's genome, better than we've had before. I'm pretty sure that they actually funded that research. I might be wrong. That might have been funded by a university that one of their people worked for, but I'm pretty sure they actually funded that with their own dollar. That's a
Is that
huge.
computational, like predictive models? Is that sort of how because they're kind of recreating the full genome from like an 80% scaffold or something? Is that sort of how it works? And they're using computer models for that, assumedly.
Yeah, it's very complicated. But when you're trying to reconstruct a degraded genome, you have to know a little bit about what you think it's supposed to look like. So
Okay,
you use a scaffolding that you already know, and that scaffolding is really nice. So if you're using other canine species as scaffolding, you can find where there are matches enough for it to hit the scaffolding, and you can see within those matches where the differences are. So it's really cool. You can see. You're going to miss everything that's so novel in that species that it doesn't match anything that exists today. You'll miss that. That'll just be noise in your data set. You won't even
right.
know that that's valuable. You'll have no way to know that that was valuable.
Which could
But you
definitely
will
exist, right?
get,
Another good point is that there's lots that could definitely exist. There could have been some distinctive characteristic of the direwolf that is lost to history.
yes, probably not that much though, because
Okay,
certainly, yes, that's possible. I mean, there are populations of humans that have extra genes that other humans don't have. A lot of these are just because of viral infections and so on that actually made it into the germline. But yeah, there's de novo genes is what we call. It's a stretch of DNA that's usually active. We know is highly active, but it's only active in one species, not another. But even in those cases, you can usually find in a neighboring species where that new gene came from. It's just, you can still find that stretch of DNA in its neighboring species. It's just not had the right mutations in it yet, or ever will, to activate it and have it become this active functional thing.
right.
Our genomes have a lot of parts you could consider to be spacers. So it's not the gene sequence that matters; it's the length is doing something usually for that organism. So the actual gene sequence is kind of noise, and these areas are where new genes can pop up. So something that was noise can become functional, and so but even that we can usually find it. When species are closely related, we can usually find pretty
Okay,
much most of their genome. So we've
okay.
been able to do a lot of cool stuff like this with reviving the genomes of animals that were extinct, even if their genome was very degraded. There's a lot of problems with it. It's very difficult. It's expensive. Um, this whole field, I mean, these people are puzzle solvers. Like these people, uh, it is a complicated mess to solve these problems. And you've got like savants working in this field. These geniuses that have figured this out. Just obsessed with math, obsessed with numbers, obsessed with puzzles. They're very smart people that are working on these things. There's errors. Nothing that comes out of there is perfect, but they've found a bunch. They funded that, so far as I can tell. They funded some really cool ancient DNA reconstructions. So that's cool. From that, they decided, I can't remember the number exactly. And it was vague. They said like 20 gene edits. And I don't know if that means 20 nucleotides that they modified, so 20 individual point mutations, or if it was 20 stretches of DNA that they modified. So maybe they modified it in multiple ways. It
Right.
was clear. I think they have a new paper where they go into more detail on what they did, but in the press release and all the stuff on their website, you couldn't get this information. They were hiding it from hiding it. They were conveniently not showing it to you, right?
Could you
They
give an
were
example on that? Seems like quite a large difference between a point change and an entire like section of gene. Could you give an example of like what the difference in that would be?
Oh yeah, it could be millions of differences. So it could be
Yeah,
huge. Like
right.
so a single point mutation is just one nucleotide in a
Yeah.
gene. A
So
gene is
one,
typically millions of nucleotides long, or at least thousands of nucleotides long. And you can modify one of those, or you could modify when you say that I modified this gene, maybe you modified 10 nucleotides, maybe you modified 10,000 nucleotides. I don't know. I can't tell from what you told me. If you say if I made 20 edits or edits in 20 locations of the genome, I can't tell you what that means.
right.
So they might have the paper out, which I should have read before coming on here and talking about it. But so far as I know, they made 20 modifications. And most of those came not from the direwolf genome. Most of them came from what we know from dog breeding. So they wanted it to have a white coat. And they were not sure. They found in the direwolf genome a mutation that they think causes a white coat. But they also know from past experiments that mutation in an actual wolf will cause blindness or vision problems.
Interesting.
So they did not want to put that mutation in their wolf. So they
Right.
went to the dog and said, how do we make white fur in dogs? Oh, it's this way, and it doesn't cause blindness. So
And that's
there's
just a known solved problem of how to give a dog white
yeah.
fur.
Yeah, there's actually
Interesting.
multiple ways to do it. There's multiple
Interesting.
mutations that cause white coats. It's easy. All you're doing is you're stopping it from producing pigments. So
Yeah.
you can break the pigment production in 1,000 different ways, right? There's many ways to do it. But so they chose the safest. They're like, okay, we have seen this. This mutation does pop up in dogs and in wolves, but it's usually linked to eye problems as well as white fur. We don't want to use that. Obviously, the dire wolf, if that is the mutation that it was using in its genome to get white fur, it's somehow compensated. And so this is we see this all the time in evolution. You'll have a mutation that's very beneficial, and so it gets selected for, but it also causes problems. But if the benefit happens to be more significant than the problems it causes, it'll stick.
Right.
And then over time, other mutations will compensate for those problems, and you'll adjust. So, and they don't know how the dire wolf adjusted. It's too complicated. You can't figure that out just looking at a genome. We're not sophisticated enough yet to do that sort of analysis. So they went with the safe bet. We're going to use the dog version. And it turns out that most of the edits were like that. They used to get the size. Dire wolves are bigger than a typical wolf. They used mutations. I'm quite certain they did not get that from the dire wolf. They got those mutations from dog breeds, abnormally large dog breeds. And so it's one of these things where you hear the news and you get excited, and then you read it and you're like, you're so disappointed. It's like,
Yeah.
I have this vivid experience when I was a kid. I was at Walmart and they just put a McDonald's in the Walmart. And there were all these beautiful posters of beautiful people eating McDonald's. And like, there's this little black girl with her Happy Meal, and she's this adorable little girl with these, her curls and ribbons tied in her hair, and her mom they're smiling and laughing, eating their food. This beautiful elderly couple that's looking lovingly in each other's eyes, eating their hamburgers. And then I look into the restaurant, and it's just, it's not the same types of people in there that
Yeah.
are advertised, And it was that's the experience of reading, of seeing their press release and then reading the
Right.
details. It's like, it's so disappointing. So disappointing.
Right. They really
And
did nail their marketing, though.
what's that?
They really did nail their
Oh,
marketing. Like Sublime getting George R. R. Martin in, who I think is
yeah.
actually on the paper. Like he's a
Oh
published
yeah, yeah,
author on the
yeah. He's a
scientific
co-author. Yeah,
paper. Like
yeah.
on the Game of Thrones throne, they look adorable. Like the marketing was sublime,
Yeah.
but they weren't direwolves. I really haven't seen anyone make a feasible argument that they are direwolves. And there's a big scandal around the hell as well, right?
The hell, yeah. Okay. So there are tests that they could do. They're saying that they made a proxy, and the
Yeah.
original group that came up with that definition of de-extinction, they wrote a paper saying, "Screw you! You did not do this. There's nothing about this. It's a proxy."
Oh, really? I
No.
didn't hear about that.
Yeah, they wrote a response paper saying, "This is you guys have just used even our horrible definition. You used it even wrong in your
Interesting.
marketing." So I'm gonna have to find you these papers and send them so that people can read them.
Yeah, please do. Yeah.
But the there was the howl. One thing you could do. We have a bunch of scientists that can tell the difference between a wolf skull and a dire wolf skull. You could do scans of these guys' skulls without hurting them. Just put them in a MRI machine. You can do it, and then you could also scan like 100 gray wolf skulls. These could specimens in museums. All you have to do is scan them the same way. Maybe scar up all the scan, have a graphic artist scar everything up so it all looks like old bones. So you can do a blind test and have all these people who are experts in dire wolves, have them sort the dire wolf and the gray wolf skulls and see where they put this new dire wolf. Are they going to put it in the dire wolf pile or the gray wolf pile? That would be a super fun experiment, and that would
Right.
really like, I challenge Colossal to do that. Like, they could really stick it to the critics like me and say, look,
Right.
this is such a good proxy that your experts couldn't tell the difference. If your
It
experts
kind
could
of
tell
feels
the
like
difference.
in like 2012 when Instagram funded all those studies to figure out if social media was having a negative impact on mental health. And then they
Yeah.
realized all the studies came back that they were, and they were like, shit, we have to bury all this now. Like if they did the experiment, it's like a gun firing in their face. And I don't know if they could recover from burying any data. If they did any study and then buried the results because it didn't support their narrative,
No,
that would tank
Colossal
their
would be
whole.
very stupid for doing what I told them to do just now,
Yeah.
because they're milking it and it's working.
Yes.
But here's the problem. And I could do this on my channel. Actually, I could show you 20 pictures of white gray wolves. So lots of gray wolves happen to be white, right? I could show you 20 pictures of them, and I could splice in there the dire wolves, their dire wolves, and no one would be able to. I predict that no one would be able to tell the difference.
Yeah.
Because you can't. You have to be an expert, and experts are saying that that's a wolf. Like, that's a gray wolf. So they say, oh, this is the dire wolves' howl. We're hearing their howl for the first time in 10,000 years or whatever it is. That's the marketing team says that. And they label it the dire wolf. They give it the scientific name of the dire wolf is on screen every time they show their new dire wolves. Then you interview the actual researcher involved, and she says, oh, no, they're not real dire wolves. They're proxies.
Yeah.
And she'll just tell you that point blank. But her Beth Shapiro is her name. She'll just tell you what it is. But the marketing team is just hamming it up. They're going for it. So I find this very frustrating because it's very much a betrayal. It's, if you got excited about this and you told all your friends and then you realized that it was BS or heavy exaggeration, that is a deep sense of, it's just betrayal. It hurts. It's painful.
I think I did. I think I fell for it and prematurely reposted it in my excitement, and then looked into it and did feel betrayed. Which obviously that's on me for not looking into it fully. But I was seeing
Well,
like
not really.
people.
You were being tricked by a marketing campaign. Like this is
Yeah.
oh, a team of people were working against you. It's not your fault. You had a team of people working against you. That's not.
It was just crazy to see scientists come out and also kind of give the same narrative. Now, I think they probably would maybe take it back now, but there was a load of scientists that I respect kind of reposting it, saying this is amazing. Maybe they were all fooled and they all retracted it, because I really couldn't imagine a very reputable scientist who's knowledgeable in this space, even remotely, hear about 20 genetic edits. Some of them aren't even direwolf edits, some of them are dog edits. And
Mhm.
then especially the howl and like all of these, like the howl, like that's clearly not the howl of a direwolf. Like nobody
Right.
could make that claim. Like, and
Nobody can make that claim because we don't know what the howl is. We know that this
exactly.
thing is howling.
Yeah.
Maybe that's a direwolf howl. I don't know. Like you can't make that claim.
So is de-extinction possible?
Well, hard to confirm ever, right? But
Yeah, because exactly. How do you even falsify that the animal, like you would have to even do it with an animal that you have extremely good records of its behavior when it existed? Because then you, like, there's always a falsification problem, and you create something, but nobody knows what it is really.
well, I don't think we could ever say it's going to be perfect unless, again, it's something that we know super well. So it went extinct while we were already documenting it really well. You could say that maybe like the Tasmanian tiger, right? That's one that was
Yes.
fairly well documented. We have footage of it, not much
Yeah.
footage, but that's one they say they're working on, by the way.
Yeah.
Uh, but otherwise, we could say with the dire wolf, you really could have a phenotypic reconstruction of a dire wolf because we have their bones, and you can look at the bones of the living ones and say, are those the same bones or not? Is that the same phenotype or not? So you could do that. You could have but that would be, the bone level. We
Yeah.
don't know what their skin was, what their fur. We don't know their fur length, their fur. They we don't know. But yeah, so you can have. There are degrees of certainty you could have. In this case, you could definitely be certain whether or not the bones are good enough to trick an expert, right?
Right.
And that would be impressive. That would be super impressive. That'd be a
Yeah.
very impressive accomplishment.
Do you
With
know
a
if their
mammoth,
plan is, are they planning on repopulating an area with direwolves?
I don't think so. I think that
Was
would
that
be,
ever the plan? This is all marketing.
yeah, I think I think what happened is the mammoth was their goal,
Yeah.
and they realized, holy cow, this is hard. And
Shocker.
look how long it takes for these things, and the animal rights issues. I think they're actually bumping up against some major animal rights issues because they
Yeah,
have to.
just justifiably so. I mean,
Yeah,
most species of elephants are endangered animals, right? So you're
yeah,
gestating an endangered animal with
and it's a
something that is kind of mammoth-like, but we don't really know how mammoth-like.
it is a 2-year pregnancy too, in most
Yeah,
elephants. So it's slow, and so it's so slow that people are going to lose interest in what you're doing, and you're going to lose funding. I think they dramatically, like, they promised too much without being able to deliver, and they did the direwolf pivot because it's a quick deliver. Like, we've got dogs, we know what we're doing. Uh, we we've been using dogs in labs for breeding experiments, for cloning experiments since forever, and that's what they use. They used a dog as the mother. I think they used gray wolf genome. Yes, they used a gray wolf genome. They used a dog as a mother, and they used some edits, they said, that came from direwolf. Most edits came from dog breeds. So and technically, the edits they did on that genome, the 20 edits, that's a huge advancement. We've never, I think the highest was 8 before, 8 edits at a time.
interesting.
And it was them who did that. It was them who had the other record. So they broke their own record. That is a major accomplishment. They deserve a lot
Yeah,
of
absolutely.
respect for that.
Yeah, absolutely.
It just,
How did they make those edits? What sort of gene editing technology are they using?
I think they're using, yeah, I would have to go back and look at the details on that. I mean, I think they're using a version of CRISPR, but it's the techniques that they're using are new to, CRISPR is one thing, but getting it to do its job without a bunch of side effects, off-target effects. It's a very clumsy structure. It'll edit too many things.
Right.
It'll edit the wrong part. Uh, and so there's a lot of technology involved, or a lot of technique involved in making sure you edited what you wanted to edit, and then getting it to work multiple times in a row. Well, how are you doing that? I think they're doing them separately and then splicing them together. So there's a lot of complicated stuff that they've figured out. A lot of difficult problems they've solved, and
Yeah.
this is no joke. That's real science.
Yeah, absolutely.
So, uh, yeah, it's what I hate to see. We're losing public funding of science with Doge and all this stuff, and I don't fully understand how bad it really is, because everyone's in science is scared. Everyone who's doing basic research has had their funding come from the NSF and so on. They are scared right now. A lot of people have literally lost funding. Sometimes it's a freeze. It's not actual funding cuts, but it's a freeze. But what this has done, people stop spending money. Like it's very difficult to get grants right now,
Yeah.
or it's very difficult if you sell morpholinos or other molecules for genetic research, for example. It's hard to sell it right now because nobody wants to spend money because they don't know what's going to happen to their grant. It's a very difficult time. What's going to happen is people are going to go more and more towards private funders, which means they have to
Yes.
impress billionaires, which means they have to exaggerate every claim they're making. And this
Right.
is going to be concept doping on steroids. It's like, I'm very worried it's like idiocracy. It's going to be like, it's science communication is going to get a lot harder.
Right.
Uh,
That's a really
and
interesting point that I hadn't thought about, because I think a lot about different ways of funding science, because
mhm,
I think there are quite a lot of problems with, the grant proposal process and peer
yes,
review and these locked gardens, walled castles of research that, me and you try our best to try and communicate to the general population, but God, if we're working, 365 days a year, we maybe communicate like less than a percentage of scientific work. It's all locked away, and maybe
right.
private funding helps in some way in the communication aspect, because it maybe gives you the money to get a marketing department. Yeah, it's a really good question.
Well, the National Science Foundation has been doing an excellent job. Every grant proposal that they accept has to have an outreach part to it and a percentage of that money
Mm,
going to outreach.
that's
And that's
cool. I
actually
didn't know about that.
that's how I've, that's one of the main ways that I've funded Stated Clearly is I would, I would find research I was interested in. I would contact the person and be like, hey, you guys have an NSF grant? You guys have outreach funding? Do you like what you planned for that? Because maybe you could just give that to me and I could do it. And they've done it. It's happened multiple times where
Interesting. I didn't even know that existed as a science communicator.
I was getting between, sometimes it's almost nothing that they can write you. But what would happen is I would do it for almost nothing. And then oftentimes they'd write me in to the grant the next time. That profit that I get from ad revenue lets me do other videos that I can't get funding for. And it lets me do more stuff with those researchers and have the Stated Casually where I'll interview them sometimes and other things. So yeah, the NSF grants have been awesome for my career. And the
That's
fact that
very cool. I actually didn't know that. I would love grants to have a sizable outreach and communication component. I didn't even know that was possible. That's a really cool thing.
yeah, and you can actually just apply for an outreach that could actually be your thing. I did apply for it, and at the time things were starting to get weird uh, at NSF. I think with anxiety around elections and so on, and so we actually decided not to even try and pursue it. There were so many hoops to jump through,
Yeah.
but the I don't know if that's all gone now. Like I don't. Everyone's terrified to spend money right now.
Yeah.
So now we have things like Colossal and the John Templeton Foundation and these other groups. They all all of these people, there's a catch. Everywhere you go, there's a catch. Money's not free, right? There's always a catch. And um, you could argue that what frustrates me about Michael Levin is actually just what you have to do now. It's just part of the game. You have to excite people. You have to phrase what you're doing. You have to think about phrasing it in a way that gets
Yeah,
people
scientists
excited.
have to be entrepreneurs.
Yeah.
Yeah.
Cell aggregates is not a cool term. Xenobots is a cool term, right? So all the haters are just hating on him for thinking of a cooler way to phrase it. Or they're jealous of his grant money or whatever. But this might just be a game that we have to play with. But for me, it's like I call this concept doping because it's offensive to me in the same way that if I were really into like biking, I would be offended by people doping and biking. It's like, yeah,
I
they're still
get it.
doing tons of hard work. It's not like you just take steroids and then you just magically win. That's not how it works.
I get it.
But I don't like this corruption of the sport. I don't like this. I see this as it's not new, but it's somewhat new corruption of science. I don't like it. It makes me uncomfortable, and I want to fight against it. So
I get it. I wonder what the other implications would be of science becoming more privately funded. Private pockets are deeper, and we do know that science in general is underfunded. I am so maybe there's some interesting areas of research that get more funding than they would have ever gotten in the public sector. Maybe because public grants have to be so detailed out, and you have to have all of your set hypotheses. You might be able to get more exploratory science done privately funded because again, the pockets are just a lot deeper, and who knows what could come from that.
Yeah. Okay.
Have you thought?
The National Science Foundation, what they do is they fund basic research, and what they want you to have it connected loosely to something that might be applicable someday. But they are the group that you go to if you just want to explore how the world works without having to invent something. Private funding is great right now for computer science. Like, man, you want to study AI or new ways that you can develop a software system that can learn without the information being hard coded. Yeah, there's tons of funding for you right now. So there's a bunch of questions that private funding gets excited about medicine, but we also see the corruption of private funding. You see a bunch of,
for sure,
in medical research, there's tons of corruption. There's tons of problems in that, but it's hard to talk about corruption in science because the anti-science crowd loves it, and they like to exaggerate it and say, oh, we can't trust scientists at all. But there really has been the increase in scientific fraud is growing faster than the increase in scientific papers, right? So the percentage of fraud is increasing, and it's been increasing for quite a while, where people are actually just publishing false data. They're actually manipulating their data. This is way different. Like, none of the people that I've talked about, Colossal, they're not doing scientific fraud. Michael Levin, he's not doing scientific fraud. There are people doing actual scientific fraud where they publish fake data.
Even this is all from a lack of funding. It's all coming from people feeling they need to publish and feeling they need to get certain results to make sure that the money keeps coming in.
It's because of competitive funding, and it even happens with NSF grants, because I think what the problem is, I mean, there's tons of problems, but science used to be a fairly small community, and people tracked reputations personally. You knew everybody in your field. Like, you literally met them face to face in conferences, and you would argue about things, and you tracked reputations like an ape tracks reputations in his little ape group in the forest, right?
Right.
You knew each other. The scientific field got bigger to where you can't track reputations personally. So we started tracking it by publications. So now we have a currency. We have money, a form of money in the scientific field, and that is publications and citations of your publications. These are the currencies that are being used. Whenever you have a new currency like that, reputation is hard to fake, but something like this it's a lot easier to fake. I can tweak a thing here, I can tweak a thing there. Okay. So now what you need is policing. We have in the financial institution, we have police. You can get arrested, you can go to jail, you can get in big trouble for fraud and with money, right? So we went from tribal reputations to a money system, and then we developed a policing system for that. We barely developed a policing system in science. We almost don't have one. And the one that we have is super, it's almost laughable. The universities are mainly in charge of policing their own researchers, but it's in their best interest to look the other way because that researcher is pulling in money for the university, and that researcher, their reputation is tied to the university. So of course you don't want to bust your own researcher for fraud. You might pull them aside and be like, don't do that. We noticed this thing, but you're not going to.
Or don't do it so in public. Don't do it so visibly.
Yeah.
Yeah, it's so silly when you have these systems in place where people are negatively incentivized to regulate each other, yet they're expected to. It's like we know a thing or two about human behavior, and we follow incentives. You know, we've seen enough examples of this. Anyway, Jon, this has been so much fun. I really want to get your thoughts, and I think we could go down another rabbit hole on science funding and stuff. That's maybe another episode, because I find that stuff really interesting as well, and I think there's so many issues at the moment. I want to get your thoughts on the future of gene editing and gene manipulation. So recently I've spoken to the CEO of Nucleus Genomics, one of the largest consumer genetics testing company in the world. And I've spoken to Robert Plomin a few times. Love that guy. He's a friend. But I'm kind of cautious and slightly worried about the genetic testing and genetic editing techniques that is coming down the pipeline 5-10 years, what venture back funding is going to do in terms of gene editing and what effects it's going to have. Is this something that you've thought a lot about in terms of what's coming 5 years, 10 years down the line?
For sure, what's coming 10 years down the line is going to be a whole bunch of ethical questions we have to wrestle with. That's guaranteed. Yeah, we're getting better at this stuff. We've had now humans that have been born with gene edits. And if we know about it in some cases, then for sure it's happening a lot and we don't know about it. Some of this could be good, some of it could be bad. The future is going to be very, very weird.
Yes.
And I think genetics is just one weird avenue it's going to take. The other, like, what it's going to mean to be a human is going to change, maybe in our lifetime. I think in particular with these computing systems that we're developing, it's really changing a lot fast. So
I would say certainly within our lifetime.
Yeah. We've already, GPT-5 was just released and it's insane. I mean, it's just, it's funny because you watch Star Trek and they have their computer is doing what GPT-5 does, right? But there's no moral implication, or there's no, it's just not a big deal. It's just their computer. It just answers any question they have. It's interesting to realize how big of a deal that actually is. I mean,
yeah,
this is changing everything, and it's changing everything rapidly. Honestly, I haven't worried too much about genetics because I don't think it's the fastest moving target. I think genetics is moving slower, It's slower. It's slow enough that the thing that's more of a concern for how it's going to change everything is computing, like what we're doing with computing technology.
Interesting. Do you have opinions on, what role gene editing should play in IVF and in embryo polygenic scores, these kind of things? When people start doing their IVF and getting their 8 embryos and getting their polygenic risk scores on all of their embryos, telling them how intelligent and how brilliant they're going to be, in all these different ways, and then the parent suddenly wants that embryo to be 2 centimeters taller or have blue eyes instead of brown eyes, or
Yeah.
maybe there's too many autistic traits in that child and that's going to cause the parents issues. And so many areas now that sound murky and disgusting, I think could
Yeah.
become commonplace.
I agree.
And I'm really curious on how people are generally thinking about it now, because as the human brain is really bad at forecasting exponential curves.
Mm-hmm.
And like
Yes.
genetics, gene
Yes,
editing
especially.
might be a 15-year problem. It might be a 5-year problem. We actually just have no idea. And I'm like,
Well, here's the reason why it's not. I don't think it's a 5-year problem. It's just because it
okay,
takes too long to make a baby, right? So, but it could be like it could be a 5-year problem for parents that are thinking about it in 5 years. But we're not going to be surrounded by super soldiers in 5 years, because
yes,
you have to
but
wait for them to grow up for 18 years or whatever.
yes, but we might be making decisions on if parents should be able to choose the eye color. Like
Yes.
even now, I find it interesting that the US have different regulations than Europe. Like you
Yeah.
can't choose sex for IVF in Europe, but you can in the US, and I find that
Yeah.
interesting. So already humans are divided on this, and it's barely been an issue.
Yeah.
There's a lot of cases where we can see gene editing as having an amazing impact. If we
Yeah.
see a Huntington's gene in an embryo, I don't think anyone would disagree in the world that we should try and rid the world of these kind of monogenic disorders, cystic
Yeah.
fibrosis. There's a lot of them that plague humanity, and they're no good. There's no use to them. But then there's lots of edge cases. Almost all of the
Yeah.
cases are weird and blurry and strange, and I don't know how to think about them. Should we be able to, like, editing behavioral traits gets incredibly complicated because things like intelligence are contributed by basically our entire genome. But
Yeah.
there probably, I suspect that in 5 years we're going to be able to do gene editing that makes people uncomfortable
Oh yeah.
around maybe things like height or eye color or obesity or extroversion or confidence or depression. There's an infinite amount of these traits that we can dissect, and we're probably going to be able to manipulate embryos in some ways to move the probabilistic bar on a few of these. So I'm curious, like,
Yeah.
have you thought about that? Like, what do you thought and thinks in general about what I said?
Yeah. Well, first of all, I agree with you. Like, that's all that stuff is coming. But personally, I haven't thought about it much more than watching the movie Gattaca. It's
Interesting.
like,
You're the genetics guy on YouTube, so I in my head,
I am
I
the
thought,
genetic guy on YouTube, but I don't focus on humans. It's not my it's just not my focus. So, but
that's
there's
true.
it's a huge mess. We already have selective breeding, and we've had eugenics in the past, and there are versions of eugenics happening today. There's eugenics is basically just the selective breeding of humans. And then there are these edge cases, because everything's got fuzzy boundaries where, well, we choose our mates, so we are practicing our own personal eugenics when we choose who we're going to mate with, right? You could say, but we're about to take it to a whole new level. And here's what happens when you give people all these options: is people everyone kind of converges on like one or two options, and then you get too much of that one thing. So we could have like
Yes.
a million of the same guy walking around. But and then the other thing is, we can't predict the future. Like the Nazis were trying to build a super race, or really, you know what a super race is. What the selection pressures of the future are going to be, and so how to prepare for that. Do you have a time machine? How are you doing this? Because this is the way that nature does it. We have diversity, and then when a problem arises, some of that group can overcome that problem. Some of the group cannot, because there's genetic diversity. And so there's one of the big problems with genetic engineering, and you see this with crops as well. So I have thought about this more with crops, not so much with people. But with crops, you have these monocultures that are then suddenly subject to, if you have one parasite that can affect that crop, and can get around all the barriers, suddenly we have a catastrophic drop in the food supply.
Famine,
So
yeah.
monocultures are bad.
Yes.
Diversity is good.
Yes.
This is what we've learned studying evolution. This is the lesson that the Nazis somehow got the opposite from this, but this is what we've been learning in studying population dynamics: is genetic diversity is good. So you're right. There are some diseases. It's like I can't imagine ever thinking that, you know, nearsightedness is going to be a good thing, right? But there might be some weird reason where the nearsightedness gene also gives you immunity to this virus that someone cooked up in a lab over there, or that evolved out of the meat industry over there. And for
They're
some
all
reason, people that are
two-sided coins,
nearsighted.
right?
What's that?
They're all two-sided coins. So, you think you're selecting for creativity, and suddenly you have a 200% chance of getting schizophrenia. Like
Yep. Yeah. Yeah.
that's one that we've probably mapped already. And you think you're selecting for height, but then who knows what that does to your immune system or your bone density or your
Yeah.
chance of getting arthritis when you're 65. And
Yeah.
I think you make a good point on the monoculture. I think once you start artificial selection, what is going to happen to the natural diversity of populations? And kind of one thing that I'm sort of worried about is traits like autism and ADHD, and I study aphantasia. These are expressions of neurodiversity which are incredibly important for humanity. But
Right.
if it becomes a trend in Californian IVF clinics funded by venture-backed companies that suddenly we don't really want autistic traits because it makes it harder on the parents, that's like a really bad thing. That's a really bad outcome. I think that's a really sad thing. And we kind of need to be having these conversations now, and lots more of them than I've been seeing, because I'm sort of thinking this is not too distant in the future with the current rate
Yeah.
of technological development. And I think,
Yeah.
like, if you don't want it to be the case that the technology arrives that we can do it, and you suddenly have VCs pushing companies to supply it as a product, but we haven't had all of these conversations around them,
Right. Yeah. Yeah. I agree. It's just humans haven't been my thing.
I love
But
that, and I love that respect. I love all your cool animal videos, and I do respect that someone's just out there covering the three-toed skink. Is that the one I watched today?
Yep.
I love that stuff.
uh, my advice to people, they're like, I'm worried about what kind of kid I'm going to have, so I'm going to go use science to make a better kid. Just like choose a good partner to reproduce with. Just like, just
I think that's rational,
it's
logical advice for
yeah.
irrational people.
Yeah. Someone you're attracted to, someone who's healthy enough to have kids. If you're a guy, you're selecting a female partner who can have kids. And if you're a female, you're selecting a male partner who can give you good kids and will probably stay with you, hopefully. I mean, these are the things that we've been doing since forever, and they work. And it's time tested, proven that if you choose a good mate, you have good chances of reproducing well. So if reproduction is what you're into, I recommend doing that. Find a good mate.
I wish people were that remotely rational, man. I think people are going to be selecting for some weird shit in the future that make no sense to us at all now. Like stuff that we wouldn't even think is desirable or admirable. Even, like trends and fads could be a thing for like choosing your child. Oh, it's fashionable now to have left-handed children. There's lots of weird stuff coming down. John,
Yeah.
you've spent a lot of time studying animals, which I love and respect, and I'm a huge fan of just sitting down and learning about why snakes lost their legs or such, and really interesting topics that you've heard. What is the most amazing animal fact you've ever learned in your life?
The most amazing animal fact. Well, what comes to mind is what I just saw recently. So it's impossible to tell you what's the best animal fact. But what I saw recently, I watched a 17-year cicada exodus. The cicadas were all coming out at the same time. I was down in Kentucky visiting a friend, and that is amazing. I mean, these things spend 17 years underground, just growing slowly and drinking sap from the roots of a tree. And then they all time their emergence to come out at the same time. So they overwhelm predators. There's just too many. You can't eat them all. And what shocked me the most, or what was, I guess this is kind of like the horror of natural selection, the horror of evolution. I saw so many that failed. Yeah, they weren't eaten, but they failed because as they were trying to inflate their wings, they shed their exoskeleton and then they try to blow up their wings because their nymph mode that crawls out of the ground does not have wings. They shed their exoskeleton, then they try to pump up their wings. They're all soft and they're gooey at this point. If another one steps on them, it can poke a hole in their exoskeleton, and then they just squirt liquid out of their side, and their wings are shriveled and deformed. And there were thousands of them that had suffered that fate. So they went 17 years, and right before getting to mate, they got stepped on by one of their buddies. And it ended it all for them. They're no longer worth, like, they can't find their mate. They can't do it.
Why on earth does it 17 years? That's so insane, isn't it?
Yeah. There's lots of debates about that, but
why on earth would, like, what? Why on earth would it be that?
Some people think that selection is. Some people think it's leftovers from glacial periods. Some people think that it's selection to not overlap with other populations, to not mate with the wrong partner. And why 17 years? It was because 15 was taken by another group. And, like, by the way, like these long periods, we call them periodic cicadas. That only happens in North America. Other cicadas do. They do variations of this. You can get kind of periodic cicada-ish things happening in other populations, but it's in the U.S. that this happens the most distinctly, where it's like 17 years and 15 years.
And these are kind of like dragonflies type thing, is that right? They're kind sort of.
They look like what do they look like? They're definitely not beetles. So don't go thinking I'm saying that they are beetles, but they kind of look like beetles in that they're roundish, fairly large insects.
Yeah.
But they're if you live in North America, you'll see them all the time. If you live in the South, you'll definitely see them all the time, because we have annual cicadas that hatch continuously. I mean, they develop for years underground, but they are hatching continuously. So every spring they'll come out, and they come out gradually. So you'll just see a slow trickle. And then we have these periodic cicadas where they all come out at once, and it's insane.
Yeah, that's got to be an insane experience.
I mean, you're walking around outside and you're just crunching, you're stepping on corpses and shed exoskeletons. It looks like fallen leaves, but it's bugs. It's crazy. Yeah.
Cool example.
Yeah. That's my cool animal fact.
Cool example, man. This has been so much fun. I really, really appreciate it. I love your videos. I love your channel. I think I should be linking more of your videos on underneath podcasts when I think that they could be valuable to listeners, because they're so well done. As you said earlier, sometimes they cost $6,000 to make. I believe it. You know, they're just so well done, so cool. Love that you're focusing on just cool, all the bizarre, weird, wonderful aspect of the animal kingdom. And I would love to focus on that. Maybe we can go, another episode, we can just focus on the animal kingdom and stay away from the stinky humans. I would love that as well. But this has been so much fun, man. I really, really, really enjoyed it.
And let me just, as an exit, Michael Levin, I apologize if I feel like I'm picking on you. I'm not trying to.
He's personally crying at home right now. Thanks, man.
Yes. I'll see.
If you made it this far, then you must at least partially be interested in brain science. And who can blame you? It is the most interesting topic in the world. Twice per week, I talk to the most amazing neuroscientists in the entire world. So if you're interested, then subscribe to the channel. Leave a comment below who you'd like me to speak to next. And thank you so much for your ongoing support.